Thank you. Now I would like to welcome Professor Kors van der Ent. Professor van der Ent is a paediatrician and professor in paediatric pulmonology and chair of the Child Health Programme. He has founded the Utrecht Cystic Fibrosis Centre, which is the largest centre in the Netherlands, and he's also the coordinator of the EU-funded HIT-CF programme. And this is what he's going to be speaking to us about this evening. So welcome, Professor van der Ent, and thank you very much for joining us.
Okay, thank you, Sarah. Thank you. Well, thank you for the opportunity and the invitation to be here tonight, and thank you, Patrick, for the awesome presentation that you showed us just a while ago. It's quite impressive to see all these developments in the genetic field. I'm going back a little bit to the clinic, and I want to talk with you about a European project in which we will try to find drugs for patients with the rare mutations. And the essence of the project that we call the HIT-CF project is that we want to match the right drug to the right patients. And I am the coordinator of the project, but many others all over Europe are collaborating in this project. Try to move forward. Well, these are my disclosures, so be aware that there is a lot of funding in these type of projects. So this is what I was being funded by. And this is to come back to something that you are familiar with, that CFTR is a genetic disease. Thank you. Well, this is what I wanted to tell you about the mutations, and you know that there are a lot of different mutations. Over 2,000 mutations in the CFTR gene have been described. And as you see in the next slide, next slide, please. That almost, well, some 85 to 90% is currently being treated with a new drug that we call Trikafta, and you are aware of that. And a lot of patients have benefited from it now during the last months. And well, we as a CF community are quite happy and enthusiastic about these type of new drugs, and we all enjoy it. But there is a considerable number of patients, some 10 to 15%, who all have quite rare mutations, and they cannot be treated with the Kaftrio until now. So next slide, please. As you see, this picture with these ducks shows that there is one being left, and that's what we do not want to do in the CF field. So we have made a project together with the ECFS, the European Cystic Fibrosis Society, and the Clinical Trial Network to work for this single white duck and to get drugs already for this patient available. Next slide, please. Well, this is something that you already have heard several times also from Patrick. We show here the CFTR protein in the bottom that is formed from the DNA in the nucleus that is transformed into mRNA, into a protein that is being produced in the Golgi apparatus, and it is moving upwards through the Golgi and is incorporated as a CFTR protein in the cellular membrane of epithelial cells, where it can open and close and where it acts as a chloride transporting channel. Next slide, please. Well, the current Kaftrio drug is actually a combination of drugs consisting of a potentiator and two correctors. These correctors are working on the trafficking and the processing of the CFTR protein in the cell and in the Golgi apparatus, and the potentiator is acting on the CFTR protein that is incorporated in the epithelial membrane, and it influences the openness of the protein. So, and you are all aware that the combination of this potentiator and the two correctors in Kaftrio are quite successful in many of the patients that are currently treated with the Kaftrio. Next slide, please. But on the other hand, and you already heard a lot about that, is that there are all types of other compounds that can influence the formation and the production of the CFTR and also the function of the CFTR protein. And on the right-hand side, you can see on the bottom the gene therapy that Patrick was talking about, but there are also other compounds like amplifiers, mRNA therapy that was also addressed, but we have read-through agents, other correctors, potentiators, and also compounds that influence alternative chloride channels. So there are quite a couple of other compounds that are already in the clinical phase, currently available, that not have reached the clinical practice until now. So these drugs are all out of the laboratory. Phase 1 and sometimes phase 2 clinical trials have been done with these compounds, but they didn't reach the clinic and the doctor-patient combination. So in essentially the question of the HCF project, the next slide please, is how can we get these alternative treatments to these patients who cannot be treated with the currently available Kaftrio on the left-hand side. The next slide, please. So the goals of the project is that we want to get access to CFTR modulating drugs for patients with rare CFTR mutations. We want to do it in a personalized way because we are talking about very rare mutations, and sometimes we're talking about mutations that are only present in 5 or 6 patients all over Europe. So we want to do it in a very personalized way, and we want to generate an easy accessible biobank of patient-related materials for future CF research. Next slide, please. And the project is really organized as a consortium, and on the top you can see the partners that are there. It's the clinical trial network of the ECFS and also CF Europe. So the umbrella patient organization in Europe is involved in the project. A couple of laboratories all over Europe are collaborating, our own laboratory in the UMC Utrecht, but also the lab from Margarita Amaral in Lisbon, and the lab from Chris de Boek in Leuven, and the hub are incorporated in this consortium. We are collaborating with all these different biotech companies that have produced these phase 1 and phase 2 compounds that I just showed you, and there are also some supporting partners in the consortium. So it's really a big consortium all over Europe that is working on this effort. Next slide, please. Here you can see the centers that are participating in this HCF project. These centers are all members of the clinical trial network, and you see that these centers are spread all over Europe, and well, we cover almost all patients within the European community. Next slide, please. What is happening in this project is that we collect patient-specific samples. And we used an organoid model, and maybe someone of you has already heard of it, but what is happening is that you can take a little piece of rectal tissue, and you can do it with a suction biopsy, and you see it here on the side, and it's a procedure that is comparable with taking a rectal temperature, as you see on the right lower hand. It's a small forceps that is intruded in the rectum, and then a very small tiny piece of the epithelium from the rectum is removed from the body. No endoscopy is needed, and the procedure is quite unpainful. So it's a little bit weird, but it's not harmful for the patient. And from this tiny little piece of intestinal tissue, we can build in the lab what we call the organoids. And you see it on the next slide. So we take the rectal biopsy on the left upper hand, and from these rectal biopsies, we can isolate in the lab the stem cells of the intestinal epithelium. These stem cells can be isolated, and we can culture them when we bring them under the correct conditions in the lab, they start growing and they form new intestinal epithelium in the cell. So they produce a sort of mini intestines. We also call them mini guts or organoids, and they grow and grow in the lab, and they can be cut into pieces and they grow further. So you can form infinitive amounts of tissue that is reflecting the entire genotype and genetic environment of the patient. So you can culture it for years and years. You can freeze it and you can defreeze it. So you can store it also in the biobank and use it whenever you need it. The nice thing of these intestinal organoids is that the CFTR protein is represented on the inner side of these organoids, and you see it on the left lower side. If the CFTR is available and if you activate the CFTR, then it starts secreting chloride into the organoid, and with the chloride, water is following. So it's a sort of small balloon that is swelling after stimulation of the CFTR protein. And what you see in the green panels in the middle and in the right lower hand is the organoids of a patient with cystic fibrosis, and you see that these balloons are all quite small. And if you start treating this patient with, in this case, lumacaftor ivacaftor combination, then you see that you get restoration of the CFTR function and that the organoids start to swell again. So the attractive thing of this is that it is quite patient specific, that you can culture them again and again, that you can store them, that you can reuse them, and that the swelling assay is fully CFTR dependent. So it's a functional measurement of the CFTR function. Next slide, please. And you can see it here. Here you see two lines, so the dark green line and the light green line. That's essentially the swelling of these organoids over time. And if you have a healthy person, and you see in the dark green line that there is a lot of swelling over time, but in the patient with cystic fibrosis, the swelling is much less. So there is a clear discrimination between healthy controls and patients who have diseases. And so we can use these models to test CFTR modulating drugs and drugs that influence the CFTR function. And the nice thing is that we have shown that the effect in vitro, so what we see as a result in the lab on the organoids, is corresponding with the result in the clinic if you start treating patients. And you see it on the next slide, where you see a nice correlation. So if you focus on the two upper panels, then you can see on the left hand the relationship between organoid swelling and lung function when patients are treated with several combinations of compounds. And on the right hand side, you can see the correlation between organoid swelling and the lab, and the decrease in sweat chloride when you start treating these patients with the compounds in clinical practice. So it's a quite predictive model in the lab for the response in vivo in the patients. Well, the HIT-CF project, next slide please, consists of two stages, two clinical stages. So the first part was that we have collected biopsies of over 500 patients with ultra-rare mutations from all over Europe. And we started to test all different compounds on these organoids. So what we do is that we match all these drugs that are available, as I showed you, with the results in the laboratory on the organoids of these patients with these ultra-rare mutations. And the second step in the project is that we select those patients who are high responders to these drugs. And you see it on the bottom that there is a difference between patients. On the left lower hand, you see a waterfall plot, and you see that there are on the left side many patients who have a high response with regard to swelling, so they are predicted to be clinical responsiveness to certain combinations of drugs, and there are also patients that do not respond to certain drugs. So the next step in the HCF project is that we want to treat these patients in clinical phase 3 trials, and then we select the patients who are high responders in the lab, and they are brought into a clinical trial, and they are really treated with these combinations in practice. So next slide. So the first trial that is going to start this summer is that we will treat over 50 patients from this group of patients with ultra-rare mutations with a combination of potentiators, of a potentiator of correctors and amplifiers, and they were originally produced by the company that was called Proteostasis Therapeutics, and we will start treatment in these patients, and we expect that they will be responsive to these patients. What we have also seen is that there is quite a differentiated response. So there are patients who respond to Kaftrio but do not respond to the Proteostasis company, and the other way around. So there are patients who are better responders to Proteostasis compounds on the right side compared to the Kaftrio. So it's really a suggestion that we should go forward with this individualized and personalized treatment approach. Next step. The next step in the project is that we are doing biobanking. As I told you, the organoids can be available for many years after the first testing. So we will test also further compounds that come up in the near future. So the next slide, please. This is what we want to do in the HCF project in the next coming years. So we are currently working on testing new and future combinations, because what we see is that there are still a lot of biotechs working on new compounds. So in this project, we can test them on the organoids that are available in the biobank. And the special thing is that we can also do industry overarching combinations, because what we see in this biotech industry is that all the biotechs try to bring their own compounds to the market, but it's very difficult to make industry overarching combinations. The organoid model enables us to test this in a really industry overarching way. The other thing is that we can approach individual patients. As I told you, there are very, very rare mutations. Sometimes some of these mutations are not fully characterized yet. So we do not know upfront what will be the treatment response of these patients. And because they are so rare, it's very difficult to perform the classical clinical trial in these type of patients. And as a project, we are in discussion with EMA to see whether in these individual patients we can go to in vitro studies followed by in vivo treatment trials instead of doing the classical clinical trials. The next step. Well, we are currently working on doing fundraising for including new patients in the biobank, because as I told you, we have now incorporated some 500 patients, but we really want to include also other patients in other countries in Europe and outside. And until now, we stick to patients from 18 years and above, because of ethical reasons. When we want to try new upcoming drugs in these patients, we should first test them in adults before we can go into children. But in the next stage, we will definitely also collect biopsies from children below the age of 18. And another thing that we are working on in the HCF project is that we want to move forward with the fair medicine principles, because what we see within Europe, at least, that the pricing of the Kaftrio is raising a lot of discussions. Some countries can afford them, especially the affluent European countries, but especially in the eastern part of Europe, many countries cannot afford the Vertex compounds until now. And it's really interesting to see a very recent paper in the Journal of Cystic Fibrosis. The next slide, please. Because there were made estimates of how many patients with cystic fibrosis are there worldwide. And currently, we have the registries from the US and the registries from Europe, and we until now estimated that there are some 100,000 people with CF worldwide. But the conclusion of this paper is, next slide, that it's not 100,000, but that there are at least 160,000 people with CF worldwide. So about 1/3 of all patients with CF worldwide are undiagnosed until now. So there's a lot of work to do worldwide. And if you calculate this, then at this moment, only 12% of these patients can be treated by the currently available triple therapy. And that's, of course, really a shame because, well, all of these 100,000 people already, many cannot afford these drugs. And of the 60,000 more people worldwide, they are also not exposed to the available triple therapy. So one of the approaches is of the HCF project that we really want to bring these drugs to as many patients as possible in an affordable way. So we really want to discuss with these biotechs to bring the drugs to the market for affordable prices, so that everybody can benefit from them. And well, the main basic spirit is shown on the last slide. You can have the next slide, please. That we never and ever will give up until the final patient is also treated and can afford drugs for treatment of this dreadful disease that we call cystic fibrosis. So thank you for your attention. And I would like to draw your attention to the last slide where I show you the website. So if you have any questions or remarks with regard to this HCF project in the future, you can always reach out to us and we will try to answer your questions as good as possible. So thank you very much.
Thank you very much, Professor van der Ent. And I think that message that we will never give up until everybody has access to a therapy or an effective medication is a very, very important one.